Good morning, children. Yesterday we discussed the structure of DNA. In this, how can we find out the length of DNA, then number of nucleotide base pairs in some species, human diploid DNA, number of nucleotide base pairs, human applied DNA, Echerichia coli cell, E. coli cell, bacteriophage πx174, bacteriophage lambda. In this, what the nucleotide base pairs are there, and the structure of DNA, what is glycosidic bond, what is phosphoester bond, what is phosphodiester bond, purines, pyrimidines. This is hydrogen bonds, how they are helping for forming of DNA. In addition to DNA, last class we discussed this information. Now let us discuss the what in this here, in these concepts here. One more thing is there, Chargaff law. If any one nitrogen base percentage will be given, how we can find out the other nitrogen base pair proportion? Then what would be there? One more thing, find out length of DNA. These two problems we solve. And same context in the structure of DNA, one more problem is there. What is the problem? Find out the number of hydrogen bonds. How many hydrogen bonds are there? We have to find out. We can say this to find out the hydrogen bonds here. Find out the hydrogen bonds in DNA. Find out the number of hydrogen bonds. Number of hydrogen bonds. Find out the hydrogen bonds in DNA. How can we find out the hydrogen bonds in DNA? Find out the number of hydrogen bonds in DNA. Suppose in given DNA, question. Suppose question. In given DNA, adenine percentage is 20%. Adenine percentage is 20%. Adenine percentage is the 20%. Then find the adenine percentage is 20%. And so the DNA length is 340 angstroms. Then find the number of hydrogen bonds. Find the number of hydrogen bonds. Find the number of hydrogen bonds in given DNA. In given DNA, find the number of hydrogen bonds. What is the question? Suppose in given DNA, adenine percentage is the 20%, and such DNA length is 340 angstrom. Then find the number of hydrogen bonds in given DNA. You can see this is here 340 angstroms. This is the DNA. DNA length means one helical you take. What is the one helical length? 34 angstroms. One helical length is 34 angstroms. So that 340 angstroms means how many helicals are there? 340 means 340 by here 34 angstroms. How many helicals are there? 10 helicals. The given DNA consists of 10 helicals. And in these 10 helicals, how many nucleotide base pairs occurs? 10 helicals equal 1 helical equal to how many nucleotide base pairs? 10 nucleotide base pairs. 10 helical equals 10 helicals equal to how many nucleotide base pairs? 10 into 10, 100 nucleotide base pairs are there. 100 nucleotide base pairs. So in given DNA, what we will find out first, given DNA length is 340 angstrom. According to this here, we know constant value. What is the standard? One helical length is the 34 angstroms. One helical length. What is the one helical length here? 34 angstroms. So in given DNA, how many helicals are there? 10 helicals are there. Each helical 10 nucleotide base pair means 10 helicals. How many nucleotide base pair? 100 nucleotide base pairs. And out of 100 nucleotide base pair, What is adenine percentage? 20%. Adenine percentage out of 100 nucleotide, adenine percentage is the 20. You know that Chargaff's law, A equal to T, G equal to C. A percentage is 20 means P percentage is also 20. Then followed by what about the G 30, then C 30%. You know that this law. But as per that, first A, number of hydrogen bonds here produced by A, the contribution of A. Here hydrogen bonds formed by A. Hydrogen bonds formed by A. A means adenine. Number of hydrogen bonds formed by A. But how many hydrogen bonds are formed? 20%. Adenine stage is 20%. 20% means out of 100, 20%. So what is the percentage out of 100, 20% means how you can see this here? This is 20 adenines are there. The proportion of adenine 20. So what about this here? Thymine 20. Thymine percentage is also 20. This is here you can see adenine is 20% means how many A's will be there? 20 A's. Here next one you have to see here T's. Number of T's. Number of thymines. How you can see total number 100 and number of percentage 20. Here what would be there? There is also T's are 20. Out of 100, A's are 20, T's are 20. Next, what about the G's? What about the C's? You can see here. Number of G's, number of C's. Number of G's. What is the number of G's? Out of 100, 30 by 100. This one cancel. This answer is the 30. And number of C's. What about the number of C's here? Out of 100, 30. Then you can see here 100. So here what would be there? C's 30. Total nucleotide base pair given DNA. What is this here? This is a DNA. One helical you take. One helical length. What is the one helical length? 34 angstroms. 10 helical means 340 angstroms means how many helicals here? 340 by 34, 10 helicals. Here, like this one helical, 10 nucleotide 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. Here, one helical equal to 10 nucleotide base pair. 10 helicals equal to 100 nucleotide base pair. So that in given DNA, how many nucleotide base pairs? 100. Out of 100, how many A's will be there? 20 A's, 20 G's, 20 B's, 20 C's. Now hydrogen bonds. A can form how many hydrogen bonds? 2. So that here, what is the number of hydrogen bonds? 22 plus 40 hydrogen bonds by A contribution. And you can see here, what would be there? 40 hydrogen bonds by here T contribution. Next one, G. G can produce. G is linking with C by using how many hydrogen bonds? 3. So what is the contribution of the 33 just 90? Here is also what would be there? 33 just 90. Total how many number of hydrogen bonds? 40 plus 90 plus 90. Equal to how many hydrogen bonds are there? 40, 80. 80 plus 90, 170. And 170 plus 90. How many are there? 170 plus 90? 170 plus 260. There are how many hydrogen bonds will be there? 260 hydrogen bonds will be there. There is 90, 90, 180, 220 plus 40, 260. Total 260 hydrogen bonds will be there. In given DNA, what is the number of hydrogen bonds? First, how we have to here calculate this number of hydrogen bonds here? Length. Here in the question, length should be given. 340 angstroms, 150 angstroms, 200 angstroms, whatever length is given. But how we can find out the number of helicals here? By 34. Length by 34, number of helical. One helical equal to how many nucleotide base pairs? 10. Number of helicals into 10, total number of nucleotide base pairs will come. But out of total number, any one base percentage is given. A is 20% or anyone. One is given according to Chargaff's law, we can find out the contribution of other nitrogen base pairs. So that A% 20 means D% also 20. Therefore, remaining 60, here it is occupied by the G and C. So G% 30, C% 30. Therefore, what would be there here? There is a 20, 20, 30, 30. Now A can pair to T by using 2 hydrogen bonds. Wherever number of A's into 2, those are hydrogen bonds. Number of T's into 2. Number of G's into 3. Why? Because G can pair with C by using 3 hydrogen bonds. Number of C's is also 3. So here number of A's in given DNA, how many will be there? 20. 20 into 2, 40 hydrogen bonds. Number of T's, this is number of T's here, 20 into 2, 40 hydrogen bonds. Number of G's here, what would be there? 30 into 3, 90 hydrogen bonds. Number of C's, what would be there? 30 into 3, 90 hydrogen bonds. Total, how many hydrogen bonds are there in given DNA? There is a 216 hydrogen bonds are. Like this, there is a choice to ask question to find out what is this number of hydrogen bonds, length of DNA, number of hydrogen bonds, proportion of nitrogen base, one nitrogen base percentage will be given, then find out the other nitrogen base percentage. Like that, there is a choice to ask questions. And this is what is the structure of DNA. Then next, what we have to do this here, structure of DNA, package of DNA. Then another thing, what is here, package of DNA. Package of DNA. This structure of DNA, package of DNA. What is the length of DNA? 2.24 meters in human being. You can see this here. Human being DNA. DNA length, especially in diploid DNA. In diploid DNA, yesterday we discussed human being DNA length in diploid DNA. How many meters are there? 2.24 meters. Human diploid DNA length is 2.24 meters. But you see this nucleus. What is the nucleus length? 1 into 10 power minus 6 meters. In terms of meters, 1 into 10 power 6. That means here 10 lakhs pods in 1 meter. But here this is the 2.2 meters. More than 2 meters length is there. Whose DNA? Human diploid DNA. This much of big DNA, hybridized gene occurs inside the nucleus. Nucleus length is very small. DNA length is very big. But the DNA arranged, where it is arranged? Inside the nucleus DNA arranged. How is it possible? DNA is packaged. This is a big length DNA, but it is packaged like this. Like this here means coiling. Due to the coiling, what would be there? It is packaging into small size, then embedded in the nucleus. But that is called as the package of DNA. This package of DNA, let us discuss here. What is the package of DNA? We can see. Package of DNA. This package of DNA explained by nucleosome model. This is explained by, how it is explained here, by nucleosome model. This is explained by the nucleosome model. We explain the package of DNA. What model was proposed here? Nucleosome model. In this nucleosome, what is the nucleosome? You can see. This is the DNA, big length DNA. This is the 2.2 meters here, like this DNA is there. Such a DNA is coiling. How it is coiling? In the nucleus, there is a proteins. This is a histone proteins. What is these proteins here? Histone proteins are there. These around the histone protein. Generally, DNA charge is negative charge. What is the charge? This is the DNA. This charge is the negative charge. But histone protein charge is the positive charge. 8 histones, they can make a ball-like structure. That is called as the histone octamer. 8 histones. What it is making here? This is the histone octamer. What is this here? This is a histone octamer. But in this histone octamer, how many histones will be there? 8 histones will be there. In histone octamer, 8 histones. But how many types here? 4 types. Types are 4 types. Total number of histones 8. Types are 4. That meaning what is here? Each type 2 copies. Here what is here? Each type is 2 copies. Each type. How many copies will be there? There is 2 copies of histones will be there. Histone octamer consists of 8 histone, but these 8 histone belongs to 4 types. In each type, how many copies will be there? There is 2 copies are there. But what are those here? Each type here, these are the types are H2A, H2B, H3, H4 histones. In histone octamer, there is a H2A, H2B, H3, H4. But H2A, 2 copies. H2B, 2 copies. Here there is how many H2As are there? 2 copies of H2A. H2B's, how many copies are there? 2 copies H2B. How many copies of H3s are there? 2 copies H3. How many copies of H4 is there? 2 copies of H4 is there. 2 copies H2A, 2 copies H2B, 2 copies H3, 2 copies H4. Total combination of these 8 histones, what has been formed? This histone octamer has been formed. But around this histone octamer, this DNA, this is coiling. Here, this DNA is coiling. Then what can be formed here? This can be called as the nucleosome. But at this nucleosome, end of this nucleosome, it can be sealed by another histone. What is the histone? H1 histone. Like this here, the nucleosome model means the wrapping of DNA around this here histone octamer, making a bead-like structure. Such bead-like structure is called as the nucleosome. Like this, several nucleosomes can be occurs in our chromatin. But next level, DNA will become a modified into nucleosome. Next year, how it is converted nucleosome? Chromatin. Next, chromatin, how it has been making chromosome. But chromosomes, where it is embedded? Inside the nucleus. So what is the organization? First, this nucleosome model, what would be their organizational package of DNA? First thing is the DNA. DNA plus histone, nucleosome. Nucleosomes are organized into chromatin. But in organization of chromatin, what can be required here? Non-histone proteins also required. In formation of nucleosome, here this is a like this. Here this is DNA plus histones. Here what can be formed here? There is a nucleosome. This is nucleosome. Here how this is called here? This is called like the nucleosome. Nucleosomes, how they can be organized here? They can be organized into chromatin. This is here. What is this here? This is organized into chromatin. In chromatin, how these nucleosomes are appears? Beads on string. If you see on electron microscope, this nucleosome appears on the chromatin network. How they can appear here? They will appear as a beads on string. Small bead is there. The small bead is there. What is the small bead here? This is the nucleosome. What is this here? First DNA. It is coiling around the histone octamer. Histone octamer is nothing but here. There is a proteins. There is only proteins here. What are those proteins here? There is 8 histones will be there. They comes under 4 types. But here these 4 types, they are H2A, H2B, H3, H4. Each one 2 copies will be there. Total histone octamer can be there. But histone, what is a histone? This is a protein. This is protein. Protein means how it is made? Amino acid. What kind of amino acids are there? Basic amino acids are there. They are the basic amino acids. What are those basic amino acids here? lysine and arginine. What are those here? Lysine and arginine. These two are what are this here? Basic amino acid. So lysine, arginine, these are the basic amino acid. These basic amino acid present in the histone protein. Combination of 8 histones, what can be formed? Histone octamer can be formed. Then histone octamer H2A, H2B, H3, H4, 4 types will be there. Each one 2 copies. Then an octamer can be formed. This is the histone octamer. This octamer, it has been coiled by 2 terms of DNA. Then end of this DNA, what will be there? H1 histone is there. Then what can be formed here? There is a nucleosome can be formed. But these nucleosomes again wrapping with the non-histones. Combination of non-histones and then what would be their nucleosome? What can be formed? Chromatin can be formed. But in this chromatin, what would be present here? There is a small beads. These are the beads on a string. These are the beads. What are those here? These are the beads on a string. What are these beads? How they can be called here? Nucleosome. And now these chromatin, how they can be converted here into chromosome. Like this. This is what is this here? Chromosome. Chromatin is organized into chromosome. So here, this is what is here? Chromosome can be formed here. So here, DNA, nucleosome, chromatin, chromosome. Like this here, organization. But in formation of nucleosome, what proteins playing a prominent role? Histone proteins. In the formation of chromatin, what proteins are playing a role here? Non-histones. Chromatin formation, what will be there? Non-histones. As well as here, then some amount of RNA is also required. Some amount of RNA is also required. Some amount of RNA is also required. Then you see, this is a histone octamer. It is a DNA. Nucleosome can be formed. Here, these nucleosomes, they can be organized into chromatin. But on under the electron microscope, in the chromatin, bead-like structures we can see. But here, next one thing, what would be there? This chromatin can be organized into the chromosome. Here, what is the diameter of this DNA? 2 nanometers. But here, this chromosome, what would be this chromosome here? 1400 nanometer. Due to the organization with proteins here, high complex structure will be formed here. This has been embedded, where it is embedded inside the nucleus. These chromosomes have been. So here, even though DNA length is much more, but it has been organized into this here short structure and here much more stout structure. But this stout structure, what would be there? Chromosome. Up to this 30 micrometers, 2.2 nanometers to how much it will be there? 30 micrometers chromosome. Largest chromosome 30. But here is so many chromosomes, 1 to 0.5 to 1 micrometer, 2 micrometer, like this here. Micrometers length chromosome can be organized here. But 2.2 meters here, what would be there? Comes under to the micrometer size. What is the micrometer? Chromosome size is the micrometer. Length of chromosome is the micrometers in size. Like this here, this DNA is organized into this here short length chromosomes here. This is here, the short length chromosome easily embedded inside the nucleus. This is what is this here? Packaging of DNA. Package of DNA is nothing but here. The DNA length is 2.2 meters. This much big length DNA, how it has been embedded in this short diameter of nucleus 10.-6 meters. 1 into 10.6 meters means 10 power minus 6 meters. That means after the 10 lakh spots in 1 meter. That much of short size nucleus, this is a big length DNA. But how this DNA become very short? In the form of chromosome. DNA length 2.2 meters. If you have to separate these, the DNA of all chromosomes, it will be there here. There will be a 2.2 meters length will come. But how it has been organized in the form of chromosome. So this is called package. The organization of what is here DNA into chromatin is called as the packaging of DNA. Chromatin is nothing but chromosome. At the stout structure chromosome, net-like structure chromatin. But what is playing a prominent role in package of DNA? There is a histone proteins are playing a prominent role.
These histones not present in here prokaryotic cell. In prokaryotic cell, in organization of or packaging of DNA into chromosome, there would be no histones. Histone proteins present in eukaryotic cell. Histones absent in prokaryotic cell. Histones not present. But histones thing is what is the major feature of histone? Basic amino acids are present in histone. What are those basic amino acids? Lysine and arginine. Now you have to see this. What is this here? In this histone proteins here, lysine arginine is there, but in eukaryotic cell there is a 4 types of histones. Those are H2A, H2B, H3, H4. These all assembled into the histone octamer. Histone octamer, it has been coiled by 2 tons of DNA. Now we are at the end of this nucleosome. What would be there? H1 histone is also there. But these nucleosomes at higher level, how it is organized here? Organized into chromatin. But in this chromatin, in the formation of chromatin, along with histone, what would be playing a prominent role? Non-histone, small amount of RNA is required. And one more thing here, this chromatin, it is looking like a net-like structure. This chromatin can be seen under electron microscope. Then the nucleosome appear as a beads on a string. How it will be appears small beads on a string. It would be appear. So here, this nucleosome, it would be appear as beads on this string. This is called chromatin. But this chromatin, this is 2 types. According to staining technique here, carmine chemical is there. This dye or color added to chromatin. Some part of chromatin, it is possessing active genes. Some part is not having active gene. The part of chromatin which has active gene and light stained, that is called euchromatin. Chromatin, how many types will be there? There is a euchromatin is there, heterochromatin is there. What is this chromatin? There may be euchromatin is there. And another thing, what will be there? Heterochromatin is there. Euchromatin and heterochromatin. What is the euchromatin here? Light stained chromatin. Light stained chromatin. This is a light stained chromatin. Light stained chromatin, what would be present here? Active genes are present. The chromatin which has active genes and light stain, that is called euchromatin. Heterochromatin means here dark stained chromatin. This is dark stain. Chromatin is the dark stain. And what genes are there? Inactive genes are there. There is a inactive genes and dark stain that can be called as the heterochromatin. Euchromatin, heterochromatin. Heterochromatin is the dark stain having inactive genes. But it would be organized, how it will be organized here? Chromosome. Now you can see this is a chromosome here. This is what is here? Organized up to this here micrometers length here. Millimeters to micrometer size is millimeter. Then what would be there? Big size, big measurement compared with this here micrometers. Like this is organization of the chromosome. One more thing is there. What is this here? Nucleosome. How many nucleotide base pairs? 200. In each nucleosome, how many nucleotide base pairs are there? If you observe this nucleosome, this is a nucleosome. It is what is here? Nucleosome. Here it is the H1, but it is the DNA. What is the DNA? It is occurs in the nucleosome equal to how many nucleotide base pairs? The number of nucleotide base pairs. Number of, this is important, right? Number of nucleotide base pairs. Number of nucleotide base pairs occurs in DNA of nucleosome. DNA of nucleosome. How many nucleotide base pairs are there in DNA of nucleosome? If you observe this nucleosome, number of nucleotide base pairs, but the DNA is boiling around histone octamer as well as expand up to the H1 histone. It would be the content of DNA equal to 1 nucleosome. So that here the DNA present in 1 nucleosome consists of how many nucleotide base pairs? There is a 200 nucleotide base pairs are there. 200 nucleotide base pairs answer. From this also here, according to 200, every 200 nucleotide base pair in the man, what would be present here? 1 nucleosome is there. Not man, in all eukaryotic cell, for every 200 200 nucleated base pairs here. How many nucleosomes will come? One nucleosome will come. So here similarly, if you observe, we can find out the number of nucleosomes. Suppose I will give one question from this point of view. You see this one here. Find the number of nucleosomes in human applied DNA. Human applied DNA. My question, human applied DNA. Find the number of nucleosomes in human applied DNA. What is the human applied DNA? What is the human applied DNA? What is the human applied DNA? 3 into 10 power human applied DNA equal to 3 into 10 power 9 nucleated base pairs. 3 into 10 power 9 nucleated base pairs human. So here one nucleosome equal to how many nucleated base pairs? 200 nucleated base pairs. But 3 into 10 power 9 nucleated base pairs equal to how many nucleosomes? Question. One nucleosome equal to 200 nucleated base pairs. Here one nucleosome 200 nucleated. 3 into 10 power 9 nucleated base pairs equal to how many nucleosomes? Question. We have to find out what is this here, how many nucleosomes. How you can find this here? So that 3 into 10 power 9 by what is this here? 200. This is 200, it will be there. So 200 means here you can write like this here, 3 by 2 into 10 power 7. Can you write like this here? These two zeros here, 2 cancel, it will become 10 power 7. But 3 by 2 means, what is the 3 by 2? 2 1 jars, you can do this here, 1 jars 2, what will be there? 1. Then 0.025 jars, so 1.5 into 10 power 7. This is the answer, 1.5 into 10 power 7 nucleosomes. How many nucleosomes are there? 1.5 into 10 power 7 nucleosomes answer. So you can see this human applied DNA 3 into 10 power 9 nucleated base pairs are. One nucleosome equal to 200 nucleated base pair. But 3 into 10 power 9 nucleated base pair equal to how many nucleosomes? But so that 3 into 10 power 9 by 200. It will be written as 3 by 2 into 10 power 7. It will come 1.5 into 10 power 7 nucleosomes here. This is here question, important question for competitive point of view. Find out the number of nucleosomes in given DNA. But the DNA content, you have E. coli, how many nucleated base pairs are there? Bacteriophage, lambda, how many are there? And bacteriophage X174, how many are there? Human being, how many nucleated base pairs are there in applied DNA and diploid DNA? Like this information you have. So according to this here, you can find out the number of nucleosomes. This is what is this here package of DNA. And one more thing, what is this set of genetic material? The DNA, this is the best genetic material. We are telling what is this here genetic material. Now you see, this DNA is the genetic material, but nucleic acid means DNA is there, RNA is there. So what is the best genetic material? DNA is the best genetic material. Almost all organisms, DNA acts as a genetic material. Few viruses, RNA act as a genetic material. HIV, polio virus, now COVID-19 virus. Retroviruses, RNA acts as a genetic material. But almost all organism, what is the genetic material? DNA. But how it is first known to us? The DNA is genetic material, now we are telling. But first, how it was invented? DNA is genetic material. There was some searches are going on to find out the DNA is genetic material. Those experiments in the searching of genetic material, some experiments were done. Those experiments were transformation experiment, Avery McLeod McCart experiment, Arshech's experiments were there, 3 experiments. One by one year, these in the invention of genetic material, what the experiments are there, those experiments. You can see here for the discovery of genetic material. Discovery of genetic material. The discovery of genetic material, what the experiments will be there. The first experiment is the transformation experiment. First experiment, what is the experiment here? Transformation. Discovery of genetic material, what is the experiment? First experiment, transformation experiment. Here, this transformation experiment, when it was done in the year of 1928. It is in the year of 1928 it was done. And where it is done here in 1928 is done. And where it is done here, the bacteria. What is the bacteria? Streptococcus pneumonia bacteria. Streptococcus, what is this bacteria? Pneumonia bacteria. Streptococcus pneumonia. Transformation in the discovery of genetic material. That means in the cell, lipid is genetic material. Several biomolecules are there. DNA is there, RNA is there, lipid is there, protein is there. So what would be the genetic material? The genetic material means it should pass characters from parents to offspring. A lipid is passing on, carbohydrate is passing on, protein is passing on, RNA is passing on, DNA is passing on, questionable. Before the Hershey and Chase experiment here, 1952, before 1952, no one knows this what would be the genetic material. So here, what this year some experiments were going on. What is the basic experiment for this year? Search of genetic material, transformation. In the search of genetic material or in the discovery of genetic material, the basic experiment is the transformation experiment. But this transformation experiment, when it was done, in the year of 1928 here, Streptococcus pneumonia bacteria is done. Streptococcus pneumonia, this is 2 strains are there. AC strain is there, R strain is there. Streptococcus pneumonia bacteria. What would be there here? There is a 2 strains. One is AC strain, another one is the R strain. Then AC strain means what would be the AC strain? Smooth strain. S strain is smooth strain. R strain is what would be there? Rough strain. R strain, what is here? Rough strain. Here, so you can see what is this here, S strain and R strain. Then Streptococcus pneumonia bacteria, it is here, there is 2 types here, S strain and R strain. S strain means smooth strain, R strain means rough strain. And smooth strain, this is harmful. Smooth strain is the R strain. R strain, it is non-harmful. R strain is non-harmful. Rough strain is non-harmful. Smooth strain is the harmful strain. This harmful strain, what it will cause here? It causes pneumonia. Its strain nature, what is its nature? It will cause pneumonia. But this rough strain, non-harmful means no pneumonia. But rough strain means, what is mean by rough strain? You see this bacteria. This is a bacteria will be there, it is bacteria. But on its surface, some rough. What is this rough strain? This is there is no production of polysaccharide code on the surface. Then how it will be there? It will be rough. But smooth strain means, what is a smooth strain? From this it is a rough strain. But the smooth strain means, what is a smooth strain? there is a polysaccharide coat is there, like this here, it will be present. They are smooth on surface of bacteria. The surface, it would be smooth. That will be called as the smooth strain. It would be rough space is there. It would be called as the AR strain or rough strain. Now you have to see this here. S strain is harmful. This polysaccharide coat produce the antigen. Those what they are doing here causing pneumonia in the body. But rough strain, this is not harmful. Now in this here, what will be there? S3 variety. S strain, what NCRT only S strain or strain is there. But in S strain, what is here? S3 strain. In this case, a quantitative point of view, useful in S strain, what can be used? S3 strain. R strain, what can be used here? R2 strain. R strain, what is this here? R2 strain. R strain, R2 strain, S3 strain. These two varieties will be there. By selection of this Streptococcus pneumonia, S3 strain, R2 strain, these two. Always you remember, no need to mention S3 and R2. Quantitative point of view, S3, S strain means you think S3. R means R2 only. So what I will mention S strain and R strain. Then by using this S and R strain, they are injecting into mice. They have observed this result. What was the result was there on injecting this R and S strains into mice. Let us see the experiment, how it will be there. This transformation experiment. You see this S strain experiment, transformation experiment. What experiment here? Transformation experiment. Transformation experiment. This is basic experiment for the discovery of genetic material. First, what is taken? R strain is taken. This R strain is injected into mice. R strain, where it is injected into mice. What is the result? Injected into mice alive, what I have? Then mice alive. This mice result, what is this here? Alive. Why is it alive? Because R strain is non-harmful. This is non-harmful, injected to mice was alive. And so there was no problem. Next, this is first case. Case 2 is there. What is the case 2 here? S strain. Case 2 is the S strain. S strain, this is where it is injected. Where it injected to mice. Then whatever the result here, mice, this S strain, small strain injected to mice. S strain is harmful strain injected to mice. Then what happens, mice, it will die. Here, S strain injected to mice, what would be the result collected? This was died. But R strain, non-harmful, injected to mice, what would be the result here? Alive result. Now, next thing here, third case 3. What is the case 3 here? You have to see this S strain. S strain, what case here? This is heat and killed. This was heat and killed strain. S strain, heat and killed, injected to mice. This was injected into mice. Then what would be the result? S strain, heat and killed, injected into mice. What would be the result? Mice was alive or die? It would be alive. Actually, strain harmful. But when it injected to mice, it should be died because it caused pneumonia. But in this case, what we did, heat and killed. Heat and killed means what would be damaged? DNA would be damaged. Due to the damage of DNA, what happens here? It cannot cause any disease. DNA is present, cause disease. But strain DNA is not there. Strain DNA damaged. Therefore, what would be there? Injected to mice. Mice, it would be alive. The case is alive. You can observe here S strain died. Here S strain alive. No problem. Why no problem? As we are treating and killing this S strain DNA denaturated. Therefore, there is no antigen production in the host cell. And for the year, mice was alive. And now you can see case 4. What is the case 4 here? R strain. This is the what is this here? R strain. R strain, what condition it has been taken? Alive condition it is taken. R strain alive. Next one, what is this here? S strain. S strain, at what condition it has been taken? Heat and killed condition. Heat and killed condition. What is this? S strain. S strain, heat and killed, R strain alive. These have been taken. R strain alive is S strain, heat and killed. Now this is injected to mice. This is injected into mice. Here R strain alive, S strain, heat and killed, injected to mice. What would be the result? Mice was here die alive. Here heat and killed. S strain nature, heat killed. R strain nature, alive nature. But this alive nature causes any problem? No problem. S strain heat kill nature causes any problem? No problem. This is no problem case, no problem case. But in these two are no problem cases. But it is injected to mice. Is there any problem or no problem? And you have to see this here. There is a problem happening. So what is the problem? Mice was dead. This result, according to this result only, what the principally proposed here? Transformation principle. As per this result, what the principally proposed here? As per this result only, transformation. Why this transformation principle proposed here? This is S strain heat and killed. This is R strain alive. S strain, whenever it heat and killed here, this DNA is there. This DNA, where it is going here? R strain. But in the presence of S strain DNA, R strain, how it will be modified here? This is that in the presence of one strain DNA can be transformed to another form that can be called as the transformation. In this last case, R strain can be transformed into S strain. So here the transformation of one strain to another strain, it would be called as the transformation. So due to the transformation only, what result we got? This was that. So what was the chemical responsible for transformation? Griffith did not know. Griffith did not know what was the material responsible for transformation. Something material from a strain enter to R strain. R strain, it can be transformed to the S strain. So S strain nature causing problem. Henceforth, what happens here? Mice was died. This principle it was called as the transformation. So the first transformation experiment who done here, the transfer of DNA from one strain to another strain, which results this strain can be modified into another strain. So the transformation of one strain to another strain in the presence of another strain bacteria, that is called transformation. This transformation occurs where it occurs here? In bacterial cell. Transformation applicable for not all cells here. But this transformation, whom it will be applicable? Bacterial cell only. This is not applicable to the other cells. You have to go and take one cell to another cell, but in the presence of other cell DNA, this cell may not modify. But what is the nature here? Transformation nature applicable to the bacterial cell. So this is the basic experiment and first fundamental experiment in the discovery of genetic material. Who discovered this transformation? Griffith discovered transformation in the year of 1928. But in the searching of genetic material, this is considered as a basic experiment or fundamental experiment it is. But in this case, he has selected two strains, R and S strain. R means rough strain. On its surface, there is no smooth polysaccharide. There is a rough nature is there that can be called R strain. On this bacteria, if it is a smooth coat is there, that is called as the S strain. On injecting R strain into the mice, there is no problem. But S strain causes pneumonia. On injecting S strain into the cell, pneumonia is caused. So here S strain causes pneumonia. R strain, it could not cause here any problem. Then here, what is the condition when R strain injected to mice alive? S strain injected to mice was died because this S strain causes the problem. Case 3, S strain heat and killed. Then injected to mice alive because as it is strain is killed, no problem. Killing nature, it is not causing any problem. But here R strain and S strain, Then R strain and S strain is DNA entering into R strain. Therefore, what happens here? R strain modified into S strain. Then it caused what it is here, pneumonia. Mice was died. According to this result only, propose a transformation. And tomorrow, okay, next class we will discuss other experiments, Avery, McLeod, McCarthy experiment, Arshech's experiment, remaining experiment we will discuss. If you have any doubt, any complications will be there, then send the message to my WhatsApp, seek clarification. Thank you one and all. Then tomorrow you will have examination. Timings, you know that 2:30 to 3:30, that theory paper. Theory paper this time I am giving 3 questions, 15 marks, and objective 20 marks. But according to this content, so that we are giving 20 marks for NEET, 15 marks for theory paper, it will be given 3 questions, 5 marks, 15 marks, then 20 objective will be given. The timing is 2:30 to 3:30, then followed by 15 minutes break. Next competitive examination, 20 marks. Okay, thank you, and